Okay, I guess without further ado, we can kick off the session. This is Imaging Beyond Visible Light. And the first speaker, everybody knows her, I guess, like Emma from Northwestern. So the talk title is Computational Color in Animal Vision. Let's welcome.
All right, thanks everybody for staying awake for this talk. So I'm coming here from Northwestern, where I run the Bio-Inspired Vision Lab, and we are really excited about the incredible diversity, efficiency, and robustness of natural vision. We are trying to basically understand the engineering principles that let these systems work so that we can steal all the sneaky bits for better cameras and algorithms. Lately, we've been thinking a lot about color. And by color, what I mean is that in the world there are these spectra, these spectral power distributions across wavelengths, and colors are going to be any low-dimensional projection of those spectra. So what I mean by that is I'll have some receptor functions, I'll integrate against those, and I'll get some small number of values, and that'll be my color. These receptors in the human eye are going to be your long, medium, and short cones. So for example, this spectrum can be projected into these three numbers, one for each cone. And similarly, our RGB filters on our camera have a similar function. We'll get three slightly different numbers, but they're both going to reflect the lovely blue color associated with this spectrum. OK, so I want to just start with what I would think of as kind of the standard model of color vision that applies across a lot of animals. So this visible wavelength, 500 to 700, we mostly agree on this wavelength range. We can go into the UV and into the infrared. I'd say we're a little bit weird that we don't see any ultraviolet. A lot of animals can see shorter wavelengths than we can. Few can see into the infrared, and it's kind of a production when they do. The other kind of basic principle that holds across a lot of these animals, we've seen a plot like this earlier today. There's not that many dimensions with a lot of variance in natural spectra. Basically, 2 to 4, maybe 5 components of the spectrum will get you a pretty good high-dimensional vector. And so sure enough, it turns out that most animals have 2 to 4 cone types in their eyes. All right, and I just want to be really clear what I mean by these receptor types in these dimensions. So for us, we have 3 cones. So we have a 3-dimensional color space. We mostly care about ratios of color, and so we can take something like a uniform brightness slice, and we get our familiar color wheel. Similarly, for a bird that has 4 color channels, it's going to have a 4D color space, and if you take one of these uniform slices, you'll end up getting a 3D subspace, and all of the colors that we can see will basically form the base of this pyramid that they have. Okay, so this is kind of the standard model of color vision across animals. There's 2 to 4 dimensions of color in the world. There's 2 to 4 cone types in the retina. There's 2 to 4 dimensions of color in the brain. And we can test that behaviorally by, say, training animals to pick between wavelengths of similar color and rewarding them with food if they get it right. So this holds together, by and large, across a lot of the animal kingdom. But we're looking for new computational principles, and so we are interested in exceptions to this rule. Specifically today, we're going to be thinking about exceptions in dimensionality. And there are some on both ends of this scale. There are a few animals that seem to access fewer or more dimensions of color than there are in the world. Okay? And so this is kind of confounding, because this core dimensionality of the signal of interest is very robust and well known. And so this leads us to ask, are receptors always identically equal to dimensions? They are in the standard computational model, but if you compute differently, they might not be. So we're going to start by looking at these cephalopods. So this is a cuttlefish. It can change the color of its skin to do behaviors like color camouflage. The thing that's odd about this is that they only have one receptor type. And so the world to these animals should look black and white. So it's been hypothesized, and there's a question mark on this slide because it is controversial among cephalopod scientists, but it's been hypothesized that perhaps they're using a chromatic aberration cue in these grayscale images to recover color and do this color camouflage. Boosting color dimensions like this is probably pretty familiar to a lot of people in this audience. When we do hyperspectral imaging, we want to collect this entire data cube, x, y, lambda. It's a bunch of different samples, and we have pretty harsh trade-offs in resolution trying to measure this with 1D or 2D sensors. You can basically trade any of your dimensions for another. You always kind of take a hit. And even then, because you're slicing your light into these very thin wave bands, you're going to have a really challenging SNR setting. So there's a lot of bad news in hyperspectral measurements. The good news is that the data is highly compressible. And what that means is that if I miss a sample, I can actually fill it in with pretty high accuracy from the data that I have left. And so in computational imaging, we move from this really harsh trade-off space to basically trading off our optical complexity, our compute, and our reconstruction performance. So our optics can be simple or complex. Our compute can be instant or tens of minutes. Here are some examples of great hyperspectral cameras made by the smart people in this room. And so we were very excited by this idea of the octopus because if it really could be boosting color dimensions, it would be sitting here, breaking this trade-off curve, right? They have a simple eye. They're able to change color quickly. And so we built a camera. It looks like this. It's just basic refocusing optics. 2 lenses, one of them moves. There's a grayscale sensor. These are all off-the-shelf parts. We do select the lenses to kind of maximize rather than minimize chromatic aberration. So as you move around, different wavelengths come into focus, and then it all gets mushed together in this blur on our grayscale sensor. We can solve this, right? A standard kind of inverse problem. I'll note in orange here, we have our physical forward model. In green, we have a data-driven regularizer. And in purple, we have your standard principal components projection. And we can do an iterative algorithm and recover this color from this signal. So here's an example of what a scene looks like. You can see it's doing this focus breathing, but it's not spatially uniform. And that's because there's not spatially uniform color, right? So if you have sort of multiple flashes, those are going to be 2 peaks in your spectrum. From this data, we take 5 blurry images. This is kind of the major weakness of this method. It is not snapshot. Snapshot's really hard. We didn't even try to solve it. But we take 5 1-second exposures, and in less than a second, we are able to compute an image that's shown here in RGB. But of course, this is a high-dimensional spectral image. So if we return to our trade-off curve for hyperspectral computational imaging, we see that, sure enough, we do end up in this cheap little corner. Now, cheap often means bad, and there's no reason to believe that an octopus needs a high-fidelity hyperspectral data cube. But when we give all of these methods the same number of total photons to work with, so this is like a reasonable indoor illumination for 5 seconds, we can look at the PSNR of their reconstructions, and it turns out that, in fact, in addition to being cheaper than these methods, we get very good reconstruction. And there's a good reason for this. It's that all of these systems, they take this inverse imaging approach where you add things to make the inverse problem better posed. You add filters, you added coded apertures, and all of these end up throwing away a lot of light. So just because they make the inverse problem better posed, at the noise level that we're interested in, throwing away the light is not worth it. So we believe that our high light efficiency is really what is driving the PSNR that we're able to recover. I was not able to compare to Todd's method because it wasn't out yet, but we're among friends here. I feel pretty good guessing. I think his would sit right here, right? Very few hardware components, very good reconstruction because of the light efficiency, but tens of minutes to reconstruct rather than under a second. That is not a data point. It's an informed guess. All right, and so it turns out that these simple focusing optics that give us the light efficiency that drive our performance also directly drive our computational efficiency. So the forward models for these problems are disgusting. They're enormous, right? We have this whole data cube. We're putting them into these measurements. If you want to invert the system, no, you don't, right? And so if we want to use any of these nice methods, we've got to play some tricks, right? The nice thing about defocus operators is that they enable a good set of tricks. So this is just kind of a cartoon. But if we Fourier transform our data and play our PCA projection games, we can actually reduce the structure of this forward model in a nice way and pull out a bunch of small independent linear systems that we can solve in parallel. So we go from a gigantic matrix to number of PCAs that you want to reconstruct by number of measurements. For us, that's 5 by 5. They grow per pixel, but that's not a big deal. We can solve a bunch of 5 by 5 inverse problems in parallel, no problem. So we started out looking at this, and we thought maybe it is possible to use an octopus-like system to have more dimensions than our receptor. It turns out that not only is it possible, but there are fundamental properties of the optics and the computation that make it a good idea. Moving in the other direction, this is much more preliminary. We don't have a camera. We don't even really have the principles nailed down, but I just wanted to give you guys kind of a flavor of how we believe that there is flexibility in both directions. And for this, we are going to turn to our friend, the mantis shrimp. So the mantis shrimp has a midband in the middle of the eye. You can see it there. And this midband is essentially a hyperspectral camera that has evolved. There are 4 rows. Each of them has these very narrow band filters. And so this animal can see a dozen color channels. Now, if you use the kind of standard computational models, you can make these incredible predictions that your animal should be able to resolve wavelength differences of like 1 to 5 nanometers. But then when you actually give the shrimp wavelengths, light sources to choose from, their actual delta lambdas are up here at like 15 to 25, which is much worse than people can do. And so the hypothesis here is that these receptors are not like general purpose, fully fledged dimensions of color space, which makes sense, right? What is this animal whose brain is literally the size of a pea going to be doing carrying around a 12-dimensional color space? That's actually kind of crazy from a computational standpoint. So our hypothesis is that there's task-specific vision going on, and probably because of those narrow bands, they're doing something like band selection, where you don't want to smooth everything out with a PCA. There's a specific spectral signal where you need high resolution, but you don't need it everywhere. And because it has this sort of stereotyped row, where like it's not like all 12 receptors get wired to each other, they show up in specific sets that specifically are like hardwired together in those sets. And so we think that this maybe reflects that there are multiple tasks that they're balancing, and so we would expect to find that different ecological needs get segregated to different parts of the midband. That's our hypothesis. And one of the great things about the mantis shrimp is that we have a good idea of some of the things that they need behaviorally. There's a hypothesis that the mantis shrimp has been living with the bomb. And what I mean by that is that these animals can punch so hard that they can easily kill each other, and they live in habitats where they compete for resources. So it's been thought that basically very long ago in their evolutionary past, their crazy colorful bodies and their crazy colorful eyeballs kind of got together to figure out who was going to win fights before they happened. Okay, so now our hypothesis becomes that these eyes and body colors together form a communication channel that we can analyze, and what they're communicating is threat. What this looks like specifically is if you are this mantis shrimp on the left, and you walk up to a burrow, and there's this mantis shrimp on the right. This mantis shrimp on the right will show off its biceps, and it's got right there a little pigment patch called the mural spot. And you will look at that color, and that color uniquely identifies the species of the mantis shrimp that you're looking at. And then you don't have to fight, because you can either say, I was actually just walking past, or alternatively, oh, you're so small I couldn't even live in that burrow, right? There's ways to communicate your size that in either direction prevent these conflicts from occurring. So this is like a long-standing biological hypothesis, but it's never been established before because people didn't have the computational frameworks and they didn't have the data. But my collaborators, who clearly don't spend enough time hunched over their computers, have been collecting point spectra across the bodies of mantis shrimp that live together in this one specific bay off the coast of Australia. And we are definitely still working on understanding what this data can tell us. But I'm going to just show you the very first, most basic thing you can ask, because I already think it's telling us something interesting. So here are 7 species that interact, that share space, that compete with each other for burrows, that communicate threat. For each species, I've reduced a characteristic spectra and body size for their mural spot color. So again, we can do all kinds of fancy stuff with this. Just the very most dumb basic thing you can do. We just want to ask if these row sets can predict the threat, and we're just going to start out with linear regression. Okay? We're doing more exciting things, but let's just start with this. And sure enough, some of the rows can predict it. We want that to happen. And also, they don't all predict it equally. That's kind of this task segregation that we're looking for. So for various reasons I can't get into, we're sort of zeroing in on row 4. But this already shows us something interesting, that is row 3 and row 4 kind of stand out as the threat predictors. And what they have in common is really deeply bizarre. So when I told you, oh, they've got this weird eye, I skated past the fact that this is a tetrachromatic, a multispectral camera in the UV alone. And the thing that seems to communicate threat most effectively is this UVB photoreceptor. These animals live underwater. There's like one UVB photon in the entire ocean. And somehow, for some reason, they're carrying around this like very metabolically expensive tissue, right? You don't mess around with retina you don't need. And yet, for some reason, they are measuring multiple UV wavebands. If we take arbitrary pairs, we see that indeed, it is really the UV combined with short-range visible that seems to be telling you what to do when you encounter another mantis shrimp. And I think that this is really interesting because we don't necessarily know what these principles are. We don't necessarily know if we're going to be able to make a cool camera out of them. But what I think we do know at this point is that UV, and in fact multi-band UV, dimensional color within the UV, is used for interspecies communication. We've known this for a long time in terrestrial environments, but it looks like it might even be happening underwater, where there's very, very little light to go around. So this is exciting, because if we could kind of intersect those communications, we could unravel them, and again, steal their sneaky bits and make cool cameras. But right now, we can't really do that, because while we know a lot about what the world looks like in multiband visible and even into the near IR, to my knowledge, we don't have any natural scenes data of multiband UV. We just don't know what the world looks like in these color channels, even though we know that interesting conversations are happening in that space. So I would really like to know. I would really like to collect that data. I don't really know how to make a project like that happen. So if you do, please find me, please help me. It's going to be awesome. All right, so with that, we started with this really basic question. Are receptors always equal to dimensions the way that they are in standard color vision? I think we've seen that this is probably not the case because anatomy doesn't always seem to match behavior. The thing that sits between anatomy and behavior is computation. In trying to boost our dimensions, we found cool new ways to do compressive hyperspectral imaging. Coming the other way, we mostly identified a data gap, but we think that perhaps in the future this is going to give us new principles for multitask band selection. And I just want to kind of finally point out that we've asked the most bare bones basic question about these color spaces, right? We haven't asked what these dimensions are doing or how they're processed or what their geometry is. We've literally just asked how many there are, right? Are they out there? Full stop. And so I think we are really just at the very, very beginning of understanding cool new color processing algorithms that have evolved to handle the world around us. So thank you very much, and I'll take any questions.
Well, thank you, Emma. Always very energetic, right? Very interesting talk. Now we open for questions. Any questions?
Hi, Emma. Thanks so much for such a great talk. I was curious, what is the resolution that you get with that defocus setup?
Yeah, so we do end up getting, spatially, we end up getting nice, sharp images at the full resolution. We do have a deep denoiser in there, so we do sharpen that up. In terms of the spectral resolution, for the results that we've reported, we have 30 color channels, but it's really just 5 PCAs, right? 5 principal components. So if you had like a really sharp, like a material with a very sharp spectral fall off, you could probably like sort of redesign your representation space to handle that. But so far, we've only looked at natural scenes. Yeah.
Cool. Any other questions?
Hi, Emma, great talk. I have 2 questions. So my first one is like, would it be unrealistic to kind of simulate some sort of synthetic underwater data set in this case, just to kind of get some preliminary idea of what could happen?
Yes, absolutely. And in fact, one of the things that we're already doing in our mantis shrimp follow-up is that as you go deeper underwater, I'm just going to show you it to you because it's cool. A very thin excuse for something I wanted to say already. Okay, so row 3 is the only row we really understand. Look at it. It's out past 300 and 700. That's crazy. There's probably no natural stimulus that kind of hits all those cones at once, but they are the wavelengths that disappear as you go underwater faster. And so these are the rows for narrow dwelling species. As you get deeper, the row 3 facets get a lot bigger. And then as you go even deeper, row 3 just doesn't develop. So we do know that that's sort of built in. But yes, we are definitely interested in kind of attenuating data in realistic ways.
Okay, got it. And then the second question is, what is the next animal? Do you guys have one in mind yet?
Oh, you know, it's got to have a lot of legs. That seems to be the trend. Great.
Okay. Any other questions over there? Yep.
It emits light. So is it maybe the case that like if they do it so fast and it emits UV light that's like the signal that they get? I don't know. This may be like a really far out theory. I'm not a biologist or anything. So.
I love it as a hypothesis. That is possible. We believe that this is to prevent that level of punching from happening.
Okay.
But I will ask my scuba diving buddies what they think of that. That's a great question.
Okay. Thank you.
I guess in the realm of just fun hypotheses, is it possible that the UV and IR detection is for depth sensing if they don't penetrate that deep?
Yeah, it definitely is. There's a really great paper by Tom Bodden basically arguing that color happened accidentally. Really, we were just seeing through murky water and needed different activities at different depths. I suggest you check it out. There's all kinds of things that could be happening.
Well, thank you so much. Such wonderful talk and such interesting. Let's send the speaker again. Thank you. Okay, so we have the second invitee speaker, Paulina Golan from MIT. The talk is about MRI miscomputation towards self-driving factor MRI. Okay, so let's welcome.
Let's see. So, you know, unlike shrimp, MR scanners see in Fourier space. So you are getting really huge variety of possible sensors. So this project has been going on for a while. Basically, our goal was to help radiologists see fetal brain. So if an ultrasound exam reveals any kind of challenge or problem, the mother gets referred to an MRI because of its exquisite spatial resolution. And she goes into the scanner. How many of you have had MRI before? Okay. How many of you have seen MRI images? Hopefully even bigger variety. Okay, great. So if an adult goes into a MR scanner, they lie still. If you need a brain scan, those of you who had it, they work really hard to immobilize your head. Why? Because what a MR scanner does is acquires one line or one point in Fourier space at a time. So any motion creates huge amount of artifacts. So in adults, you basically bolt their head to the table. In fetuses, you can't really do that. So while we want to acquire images and so the image in MR is a 3D cube filled with tissue, right? And so if you want to see a fetal image whose cross sections look like that, so to orient you, the leftmost one is a cross section like that, and it looks like a blurry version of an adult scanner with much less gyration. For those of you who have seen those, this one, the middle one is an axial slice, so it's a cross section like that. And here, the fetus is looking to that side of the room. The reason why you can't see any background, there is mother around this fetus, and also there is fetal head, fetal body. We basically sort of masked out everything else so that you can focus on the head. Instead, what happens is this. So First of all, the leftmost one is not upright, so it makes it difficult for radiologists to see. Think about all those experiments that you have seen where you're supposed to recognize faces upside down. Basically, this is what radiologists have to do confronted with these images, right? They have to tilt their head to be able to detect features associated with problems. And the other two are completely useless. So what happened there? The baby moved so much inside the scanner that there is huge amount of difference between the slices in what they acquire. They're not actually coherent slices that you can stack together and call it a volume. But instead, what you're seeing is sort of jumbled up picture where every slice got some cross-section of the brain, but they're not necessarily going front to back. So here's the video of one of these. Oh, okay. So here you can see the mother and the fetal head and the baby. And basically in every frame here, the baby is looking at us and then sideways, at us and then sideways, and then it moves and so on. And so instead of planned slices that go like this, you end up with this fan-like stack. And the radiologist has to look at it and say, is it okay or are there problems? And so what happens in reality is that a technologist who acquires the scanner and the radiologist are in constant communication while the scan is happening. And the radiologist said, nope, this is not good enough. Another stack. Nope, not good enough. Another stack. So our data downloaded from Clinical Archive shows that many mothers stay in a scanner for 40 minutes before clinical quality images are acquired. This is in contrast to your 5-minute MR brain. So 40 minutes to acquire 5 minutes worth of data that the radiologist can read, and some of them the mother just quits because it's just impossible to catch the baby not moving too much. So we're gonna solve this, and so this is where computation will interact with MRI. In particular, our solution is as follows. So the pink slices are the slices of MRI that we would like to acquire. We will insert a really low energy cube volume, so acquired by MRI. Because it's very low energy, it has to be low resolution and low contrast because there isn't really much of activation happening there. But we'll tune it so that it's fast enough to fit between the slices. I'll tell you in a second what I mean by that. And it has just enough information about the baby's head location to identify it and dynamically reorient the imaging plane of the next slice to follow what we wanted to acquire as opposed to where the baby moved. So in some sense, we're chasing the head of the baby and dynamically changing the slices to be relative to the baby's head rather than relative to the global coordinate system of the scanner. Okay, what do I mean by fitting in? So it turns out that because of the energy constraints on fetal imaging, which are pretty strict, there is 1.5 seconds of dead time between every 2 slices, because enough of energy gets deposited in acquiring each one of these slices, you have to cool down before the next slice is acquired. And so the 1.5 seconds is to acquire the image, estimate the fetal head pose, and tell the scanner to change what it's doing. These volumes take 1 second to acquire. They're about 5 millimeter isotropic resolution, and the contrast is terrible. Now, you see there are black strikes through the volumes. That's real, and it's called spin history artifact. It turns out what happens is that when you activate the slice, excite the slice in MR terms, to acquire it, it takes the spins flip, and then you watch the relaxation of them to the steady state. And it turns out they take long time. So what you're seeing as streaks, black streaks, is the previous slices. It's a shadow of the previous high energy slice in these volumes. So not only they are low contrast, they're also stripy. Okay, so our approach would be to train a neural network, because we roughly don't know of any other solution that is fast enough, to figure out the fetal head pose and transmit that pose to the scanner. And so I suspect that many of you say, I could do that. So the challenge is, and so we trained a network, and the architecture of it, I'll mention a couple of details, but really they're less important than the overall strategy. One challenge, so there are several kind of interesting items. One of them is we didn't have a single volume like that, the navigator volume to start with. So we can't train on navigator volumes for which we know the ground truth. There aren't any volumes, no ground truth, and so on. But what we discovered is that the contrast that the navigator volumes are using, this low energy, low contrast data, research scans usually use this contrast to acquire much higher resolution, much better images. So we'll corrupt the high resolution images to look like navigator volumes and train the network to work on those. The other subtlety, so we find the head, figure out its pose. The other subtlety is that the output of this network is essentially 3 vectors, right? It's where the fetal, so we have a coordinate system that is following the fetal head. So because the outputs are vectors, we basically ended up using what's called equivariant architecture that knows that 3 channels are not just 3 channels of scalars, but they're actually vectors that have to move consistently as I move the input. So that's one detail of it. And the other one is that at this resolution, the baby's head looks like a football in a sense that it's completely symmetric, maybe slightly elongated in one scale. As a result, there are natural symmetries to the object. And if you code those symmetries in, you get better performance. So for those of you who are doing this kind of stuff, it might be useful. So what we found is that the best representation was to represent up and front to back vectors as vectors, but the vector that goes left to right is what's called pseudo-vector. The term comes from physics, and there is a mathematical quality, properties of it. The bottom line is that a pseudo-vector specifies direction but doesn't commit to whether it's pointing this way or that way. So it requires modifying the loss function so that it says, I don't care which way it's pointing, I just want to know in which direction it's going. And it turns out you get more accurate estimation this way than committing to the vector, figuring out you got the wrong direction and flipping it at the end. And then we do SVD. It's a standard trick to actually orthogonalize it. Okay, so this slide is dedicated to our CVPR reviewers. We do. But what the radiologists care about is actually, so you acquire the slice stack. How good is it? I wanted the slice stack of perfectly parallel slices in a specific orientation. So we measure what we call obliqueness, meaning the dot product or the degrees between the normal vector you wanted and the normal vector you got, as well as an offset, which is the distance from where you wanted to be in a slice versus where you got. And we dramatically reduce the mismatch between what you wanted and what you got. This is all in simulations so far. And the way we simulate it is by looking at those studies and figuring out trajectories of how the babies move and simulating, using that real physiological movement to say, suppose I was acquiring the data while the baby was moved the way I saw in that study. What would the errors be? Coverage is another important measure. Basically, this is 3 subjects, 3 cross-sections like I showed you before. Yellow means that you saw that voxel once. Green means you wasted some effort because it means that more than one slice saw those voxels. So in some sense, it's not a big deal. You just wasted time. The white means no slice saw that voxel, meaning that if there was a lesion in a baby's head there, the radiologist has no way to see it, no matter how nicely your diffusion model works as a post-processing, right? Because that lesion was never seen by the MR machine. And so you can see that using this tracking, it's sort of not surprising, it's a sanity check. Using this tracking mechanism, we get much more regular coverage. We waste time sometimes, but at least it's more regular. Okay, so when we got to this point, we said, okay, let's scan some live subjects. So the red is the scanner, and it acquires the slice and the navigator scan. This is all done. I basically bought the most expensive laptop I've ever paid for because it had really good graphics card. That's what I wanted, right? I wanted the GPU. And so Rami, who's a graduate student who's leading this, has the fanciest laptop in the world. In our group. And so she has a USB cable that she plugs into a MR scanner. And so that cable, basically, on that cable, goes the navigator. we run our little network, we figure out the pose, and we append that pose to the prescribed pose that I want, prescribed imaging plane that I wanted to acquire. So one way to think about it is there is a ticker tape of prescribed slice planes, and I augment each one of them with the estimated pose so that it's in the coordinate system of the baby's head rather than the global coordinate system. The same lovely reviewers also said, well, you haven't really shown that it works in real babies in a MR scanner. And what I wanted to write in a rebuttal is, well, at that point, it's not a CVPR paper, it's a Nature paper. But Rami talked me off that ledge. Anyway, so you're one of the first ones to see this live. We actually went into the scanner and acquired it on real babies. So far, we've acquired 9 babies. The video on the left shows what it does in a standard way, where it doesn't pay attention to the motion of the baby. And the prescribed slices want to go like that. And the video on your right, shows what's happening here. So I'll let it run a couple of times as I talk through it. But basically, here, one thing to notice is that in our sequence, okay, here we go, the standard sequence. So the baby is looking at us, and you can kind of start looking, oh, it looked to the side. Did you catch this moment? It looked to the side again. You can see the eye and the brain is shown in this orientation. So in this video, twice, the baby just said that, did that. And so in the standard frame, you've lost that data, right? You didn't acquire the right slice. But in addition to that, there are these streaks going through. And that's that spin history artifact. Because the previous slice was like this, but the baby looked like that, and so you get streaks. Now, and so watching these is sort of fascinating, maybe for me. I don't know. So let's watch what happens with our acquisition. The baby is kind of scarily staring at you because it's a slice that goes like this. And so here, we show it in a stabilized frame relative to the baby's head. And so the mother is whooping around kind of the baby. Of course, that doesn't happen in the scanner. We're just stabilizing the video, right? The mother is completely stationary, and we're tracking the baby. So far, we've acquired 9 babies. Each one of them taught us something about misconceptions that we had going in. So we actually changed the algorithm. Some of it is just coordinate transforms. Oh man, this is 4 coordinate transforms that have to actually talk to each other. So once we fixed that, then there was an issue with the field of view and so on. So basically, every baby so far has taught us something. I think we're getting to the point where we're just gonna acquire babies until we don't change our code, and then 10 more, and then we write that paper. Anyway, before I conclude, I want to thank my fantastic collaborators, both at MIT and at Boston Children's Hospital, and of course, generous funding. What I showed you today is that combining physics of the scanner with the computation that has to run on a GPU so that it keeps up can help us really change how medical images are acquired. The amount of adrenaline I've consumed in this project is unparalleled by anything else that I've done. We have demonstrated stable results on simulations. Now it's show time, and we are acquiring about 1 subject a week, and hopefully the sequence will stabilize and we'll go actually acquire real data. Our hope is that my vision for this is that there is a controller that sits there that knows everything that happened to that baby. You know which slices got acquired, which ones are still hot so that you can't acquire nearby, and makes a decision about which next slice to acquire rather than the ticker tape I was showing you. And then we can really help radiologists understand what's wrong with their patients, and then treating physicians help them with therapies. Thank you.
Open for questions.
Oh, no, I don't want to run. Have you thought about how you'd extend this to like mothers with twins or triplets?
Oh, yes. Our other project is actually on twins. And it's interesting. So let's see. Older babies are easier than younger babies in some ways, which is that they get stuck. because they're big and don't have much space to move. And as a result, when I show this to my other collaborators who only work with 30 weeks on, they're like, what's the big deal? They don't move. But then I talk to my collaborators who work, so remember I said in ultrasound, if something is wrong, that happens at 18 weeks. At that point, they're doing acrobatics. So with twins, it's interesting. Computationally, it's harder, but on the other hand, they get stuck faster because there isn't as much space. It's a very interesting, very challenging problem. We can't even disentangle them. Like, we can't robustly, we've been working on this, but basically, we can't robustly tell which limb is what to the point where we can deploy it, but clearly, that's the next thing.
I had a question with the artifacts you were having with the striping.
Yes.
Would those be able to be any kind of cues for the orientation, or is it just too, like, it's just too far back?
No. This is, you hit it on the nail. So one evidence is where the head is. The strike tells you where the previous slice was. So it's not useful, at least in a naive implementation, to predict the future, but it's useful to predict the past. Why is that useful? Because if you want to know where every slice is and what you've acquired and what you didn't, you want to know the past, even if you made a mistake in the past. So the right thing to do it, we have a really talented undergraduate student who is from Caltech, actually, spending summer with us, working on the problem of figuring out where that artifact is and correcting past mistakes, basically. Yeah.
Any other questions from the audience?
Thanks. Interesting talk. I was wondering, would any complementary modalities help here for getting head pose, for instance, 3D ultrasound or ultrasound while you're doing the MRI?
Yeah. It would be interesting to know. So I know that what people do for adults, actually, I have some colleagues at Mass General Hospital who put a camera in and watch the face move. And based on that, the brain is basically rigid body, sort of attached to the face. You can model it sort of like it's a fairly accurate first order model, right? Here, I just don't know. So building MR-compatible ultrasound is not such an easy thing. We keep talking about sort of what could we bring to the sensors. It's an excellent idea. But since I have dramatically less sensor building capabilities than I think anybody in this room, I keep thinking about how I can help with computation, but there's no question that a little bit more sensing might be useful. One thing we have thought about is actually modifying the MR sequence itself to put in, you can sort of watermark it for pose. The problem is then you need to go into a clinical scanner and tell them to completely change the sequence. And in my experience, that's a non-starter, at least until you demonstrate dramatic improvements or dramatic benefit. So we are going through this route of sneaking it in when they're waiting anyway. Sorry, when the machine is waiting. I mean, it's all automatic, but basically that's viewed as dead space. And so we're sneaking in and acquiring just a little bit more and computing in that time.
Great. Thank you so much. That's wonderful talk. Let's send the speaker again. Thank you. Okay, our third speaker today. So we have like Mark from Westman Institute of Science. Very interesting topic, vision from vibrations. When motion becomes meaning. Let's welcome.
Okay, hello everybody. So at a high level, as you all know, computer vision is about sensing the physical world and extracting some meaningful semantic information from the raw visual signals. And in the past several years, I've been fascinated with the question, what if we could do computer vision, but with a camera that can see sound? Okay? And in today's talk, I'm going to tell you about my journey towards answering this question. But first, what do I mean by seeing sound? Well, to understand what I mean, let's think about the key differences between our senses of sight and hearing. Okay? So first, the information in vision is mostly spatial, right? So you can look at this single frame from a video, and understand that there are two guitars in the scene. But in hearing or a microphone, the information is mostly temporal. Right? You can hear that there is more than one guitar from this one-dimensional signal that I'm playing to you. Secondly, vision gives us information directly from the surface, right? Neglected any atmospheric effects. But with hearing, you have no choice but to integrate the sound from the entire environment. So when I say scene sound, what I mean is the ability to measure vibrations remotely at the surface at multiple spatial locations at once. Okay? In this scene, by the way, if we had that ability, we can bypass the camera microphone, which records both of these guitars, and listen to each guitar separately, just like we can see them separately. Okay, so back in 2022, we built the first prototype that could efficiently capture surface vibrations at multiple points at once. And we showed some really cool applications like recording multiple instruments, like you just heard, recording Abe's voice from a chip bag, and even visualizing the object's vibrational modes. But in this first project, the camera design and the vibration extraction algorithm did all the heavy lifting. And once we extracted the vibrations, there was really nothing left for us to do but just to hit play. Okay? So the question now was, can we use these vibrations not just to replay sound, but to infer some higher level information about the scene? And if we think back on standard vision, right, one of the most basic things that it gives us is spatial localization. So can we localize something with our vibration camera? Now, in a second, I'll show you what we can localize. But before that, let me just briefly remind you how we measure these vibrations in the first place. So to capture vibrations at an object surface point, we're going to illuminate that point with a laser, and then defocus our camera to image the resulting speckle pattern. And you all know what speckle is after Grace's great explanation. Now, the 2D shifts of this pattern in our video correspond to the 2D axis tilts of the surface at the point that we're measuring. And this is how we amplify these tiny vibrations. But the problem is we want to capture these vibrations, these speckle shifts, at high speeds to do anything meaningful, and the regular camera is simply not fast enough. And that's why our 2022 prototype actually combined video from two slow cameras to compute these vibrations at much higher speeds of up to 63 kilohertz. Okay, so back to the question. What can we localize with our vibration camera? So it turns out that we can localize impulses on surfaces, like the impulse created by this ping pong ball if we drop it on the table. Each such impulse, and every time you bump into the table, you create this elastic wave, which we can sample with only a sparse set of points on the surface and use the measured vibrations to estimate the impulse location. And then we can do things like localize this ping pong ball as it hits the table during a game, while our camera is only looking at the vibrations from below the table. And we can potentially localize a person walking on the floor from the vibrations their footsteps create. Okay, so far, vibrations gave us sound and spatial localization. But can they give us something even more semantic? Okay? Like the hidden property of an object. So take a look at this scene. Okay? It has a bunch of containers. Now, from a functional perspective, right, the defining property of these containers is not their color. It's not their shape. It is how much liquid they contain. That's what they're for. Okay? In standard computer vision, strong as it is, can tell you what these objects are, but can't tell you this important intrinsic property. of what's inside them. So maybe we can use vibrations to answer this question. Now, the intuition for why the answer is yes should be clear to anyone in this room who likes wine, okay? Because if you like wine, you know that if you take a wine glass and you fill it with different levels and you give it a little tap, you're going to hear different tones. And the reason you hear this nice clear tone is because a wine glass is a very special object dominated by one vibration mode. But if you try to do the same thing with something else, like this milk carton, you're going to see a much more complex and messy frequency response. Moreover, there are even harder things to sort of analyze. For example, there are containers that have this insulating layer that creates very similar frequency responses for different fill levels. Also, there are containers that should be exactly the same, but have different frequency responses due to manufacturing differences. So our task is to figure out how much liquid is in all of these containers without ever touching them. So for this task, the dual shutter camera that I mentioned before is not enough, because it only gives us sensing on a line, and what we want is to measure many containers at once with multiple points per container. So for this paper, we had to invent a new speckle vibrometry camera, where the key hardware innovation is that we go from a one-dimensional sensing into 2D. So how does this camera work? Well, we use a slightly faster camera to image this entire 2D grid. And at full frame resolution, the camera is still too slow, okay? But luckily, this camera allows us to define multiple short regions of interest that are going to intersect every speckle row. And then we can read out only these regions at the desired high speed. So from each tiny little speckle rectangle, we can compute the 2D speckle shifts efficiently and robustly using a custom GPU-implemented method. OK, so this is our new sensing. This is our new camera. Now let's talk about the processing. OK? Now on the processing side, we take the vibration signal from each point, convert it to the Fourier domain, and then we feed it into our vibration transformer. Now this transformer has two stages. The first stage analyzes the spectrum at each surface point separately. And the second stage basically combines the information across all the points. And on the network output, we have both the container type and the predicted liquid level. OK, so we have a camera, right? We have a computational architecture. All we need is data. So to gather data, we build this nice rig and capture vibrations for various everyday containers, for multiple liquid levels, while exciting their vibration using the speaker array, playing various audio files. And we tested on a bunch of validation categories. So for example, we only have 1% error for containers seen during training but tested under new speaker positions. About 9% error for new instances of known container types. That means that we sort of trained on 5 Coke cans and tested on the 6. And we also generalize reasonably well to unseen liquid levels and even to ambient sound excitation. And in the paper, we have additional ablations and analysis that you can check out. OK, so now we have a new camera that can sense many points in 2D. And we've also shown the power of combining this sophisticated processing with the raw vibrations that we have extracted to get some higher level semantic information from the scene. Okay? We've shown this concept. But you know what? Before we get ever more semantic, right, let's stop for a second and ask ourselves, are we really done with the sound extraction task? Right? Is it solved? And the answer is not quite. And let me explain. if you look at the type of objects that I and other people are using for sound extraction, you'll see that most of these are actually easy cases. So what do I mean by this? So most of the objects here are active objects that generate their own vibrations. So these are basically speakers. While the passive objects that were used in the past, right, these are our visual microphones, were usually thin, lightweight, and highly responsive objects like this chip bag. But what happens when you try to use something else as your visual microphone in the scene? Well, let me demonstrate this with an experiment. So let's take our trusted chip bag and extract its vibrations using our newest system at a single point. And now let's just pick one of the access vibrations and play it as is. Right, that sounded pretty good, like a noisy microphone. But now let's repeat the exact same experiment, but replace the chip bag with this drum head. Now we get this. That's not so good, right? Even though this drum head, right, is still a relatively thin membrane which is free to vibrate, right? It's not a concrete wall. And yet you can hear that it attenuates the high frequencies much more aggressively, and it also imparts this resonant timbre to the recovered sound. So what can we do? Are we destined to look at chip bags for the rest of our lives? The answer is from this, okay? Remember that a new camera can actually capture many points on the surface that we are looking at, okay? So can we somehow cleverly fuse this noisy signal from all of these points into a one signal that has a significantly higher quality? Can we do that? Of course, I wouldn't be telling you if the answer wasn't yes. So here it is. Let's hear the single point recovery versus our recovery side by side to really appreciate the quality improvement. And I know what a crowd like you thinks, right? Are you just averaging all these points together? No, because if you average all these points, you get an even worse reconstruction. So you can see, averaging doesn't work. We have to do something more clever. So to fully understand how clever our solution is, we need to go carefully through a bunch of math. And it's very late in the day. We're not going to do that. Instead, I'm just going to try to give you the intuition about our solution. So what our camera measures corresponds to the spatial derivatives of the surface at the 2D grid points that we're measuring. Okay? And to fuse all these points constructively, we need to know the phase and amplitude relations between all the point signals. So in this example, I picked 2 points that have the same amplitude but the exact opposite phase and the x-axis vibrations. So if we were just to average them out together, they will cancel each other out. The problem is this relationship depends on the frequency. So signals can align at one frequency but be misaligned at another. So intuitively, what we want to know is something like this beautiful relationship between all our points, but for all possible frequencies in our signal. So in the paper, we do this implicitly by deriving a physics-based forward model that connects the surface vibrations that we measure to the seen sound via the object's vibrational modes, which is a special set of frequencies and vibration shapes that span the object's vibrations. And now, of course, once we have the forward model, we can recover the seen sound by inverting it. While the modes which are the key component in this solution, are estimated from the raw vibrations. So that's the gist of our solution. And now let's see some more results. So here is a result from a picture frame captured with a 10 by 10 speckle grid, single point versus our recovery. Here's this metallic trash can. Here's a guitar. And by the way, unlike prior works, this guitar is not being played. It just reacts to the sound in the room. All right, and finally, here's this nice spherical balloon. Okay, and we have a lot of other objects and results to look at the project page. Okay, so have we finally solved the multi-point sound recovery problem? So to answer this question rigorously, we also developed a calibrated recovery baseline that uses a known reference sound in the scene to directly compute the filters that map the scene sound to each laser point. And then once we have these filters, we can recover a kind of best case scenario for new unknown sounds. So let's hear the difference. So as you can hear, the calibrated recovery is slightly better, especially in recovering the higher frequencies, which means that there's still room for algorithmic improvement. Okay, so to conclude, I've shown you a couple of examples of various vision tasks that we can do with vibrations. But I think the most fundamental question that we're trying to answer with this research is what more can these vibrations tell us about the world? Okay? So for example, if you think about the vibrations of this closed box, right? Can we use them to get a text description of what's inside this box? Can we use them to generate an image of the box's content? By the way, all these questions that I'm asking and everything you've seen today, right, these are things that we already know that we want to ask because they stem from our human intuition. But because vibrations are not intuitive to us, right? They're like molecules. We don't really see them. The more exciting possibility is that they can reveal some information that we don't even know to look for yet. These are the unknown unknowns in this research. And with these exciting prospects, our journey continues. Thank you.
So what object gives you the best reproduction and why?
So the best object is a chip bag.
Best is a chip bag.
Yes, because it is highly responsive. It's almost made from the same material that a microphone membrane is made.
Right.
So that's the best. But we have this kind of the saying that if it knocks, it works. Like if you knock on something and it sounds like crunchy, kind of high-pitched, it will probably give good reconstruction. But if you take like this chair and you knock it and you don't hear anything back, it will probably not.
How about the diaphragms within a speaker enclosure, which are also at multiple frequencies?
So the diaphragm within a speaker, I mean, that's an interesting object, but it's kind of contrived, right? Like the point is just to look at something in the room as is and see how that works.
Yeah, so I guess extending my question actually is similar to the previous question. Could you combine different objects in the scene to get a better reconstruction? So like the chip bag and say like a bass drum, so something that's
Future
like
works.
yeah,
Yes,
okay,
we
yeah,
haven't
yeah.
done that. It's a task, laying on my desk for sure. If you look at a scene where your speckle vibrations come from many objects, the task of combining them in some interesting way, that's a great task to follow.
Yeah, thank you very much for this very nice talk. I have a very basic question about the basic measurement principle. So you are looking at the shift of the speckle patterns. Under some circumstances, the speckle pattern might decorrelate. And then you cannot, or it's actually then very hard to actually evaluate the shift. Do you have any assumptions about the vibration modes or the vibration amplitudes or so, or do you have a trick to still get some data from that?
Yeah, thank you. That's a good question. I'll give you a basic answer. Okay. So the speckle can decorrelate, but what we do is we compute it frame by frame, and these frames are at a very high frequency. So if you think about your speckle being decorrelated over seconds, that means nothing to us. That will be some change that will be almost transparent. It could happen, for example, that if you are capturing the speckle at a low frequency and the membrane is whatever you're looking at is moving very fast, you would have some decorrelation that could actually sort of break your sensing. But at the regimes that we are working, because we are working at very high frequencies, we're sampling this shift at very high frequencies, this would not likely to happen at all.
It's imposing a constraint on your vibration frequency, basically your camera frame
It's
rate.
it's more like if you sample 60 times a second, it might happen. But if you sample 60,000 times a second, the shift is going to be, the decorrelation is going to be so small, you're not going to notice it at all.
Thank you.
That's a good question.
Great work. I had a quick question about the work where you guys have this closed bottle and you try to predict the level inside.
Yeah.
I think you guys accounted for manufacturing defects, different materials and stuff. Are you guys assuming that the liquid in the bottle is a constant viscosity? Because
yes. Yes. In this paper, we only used water, but extending to other liquids would basically require extending your data set.
Okay, got it.
And I would think that I can hypothesize that this will work.
I have another question. If you had 2 bags of chips, could you do stereo? And that's a lead up to, if you had a bunch of bags of chips, could you do a spatial reconstruction of a sound source that's inside the room?
Yeah, that's a great question. First, if I had 2 bags of chips, I would be much fatter. But yes, if you had, the more chips you have, you basically have good microphones in your scene, and you can kind of almost create a microphone array with these bags of chips that could be used for various tasks. So this is a bag of chips is closer to an actual microphone in its quality. So putting lots of them in the scene will give you a lot of virtual microphones.
So I guess here when playing the shift as audio, it kind of assumes that the amount of shift corresponds linearly with the sound pressure. But are there situations where the materials might deform non-linearly, that deforms linearly to a peak and then it settles out? And does that cause distortion?
I cannot answer this, I'm not a material scientist, but we have not encountered any non-linear, a material that reacts non-linearly to that linear sound. So all our models and experimentally, it's always some transmission function, right? Some frequencies get squashed, others get amplified, but no nonlinear. So I'll tell you another thing. If you think about the analogy of a speaker, your speaker becomes nonlinear when you crank, right, the volume to 11, right? So basically, the motion becomes distorted. And this doesn't usually happen with vibrations because the motion is tiny, right? It's just reacting to your voice. There's nothing there to make it, vibrate violently. So not likely.
Thank you. So let's have all the discussion later. Seems like we have a lot of questions, but don't worry, we have enough time for the reception. Okay, let's send the speaker again. Thank you. So we have 2 ICCP papers left for today. Here. So the first one, we're going to have the revealing subtle heat flows, all the raw loss, using microbolometry videos, very interesting as well. So the presenter is Mani Ramanagopal from CMU. Let's welcome.
Yeah, hello everyone. My name is Mani Ramanagopal, and I'm excited to talk to you about revealing subtle heat flows all around us using microbolometric videos. If you're wondering, microbolometers are basically cheap thermal cameras. You've probably seen thermal cameras with thermal images, and they're already commodity sensors, and they are used for a wide range of applications. In many of these scenes, what's common is that these are all high contrast scenes where the object of interest is anywhere from tens of Kelvin to hundreds of Kelvin above the background temperature. But let's think of a scene closer to home, say the study room. Perhaps the hot coffee mug or the open candle flame would be of bright targets in the thermal spectrum. But I want to draw your attention to this plastic bunny sitting on the windowsill. We actually staged such a scene, and we captured a thermal video of it. And this is how it looks in a linear color map. It almost looks like nothing is happening in the scene, and that's because of this linear color mapping. But if you were to use contrast enhancement, you see a lot more of the details as well as the noise in it. This specific thermal camera has a sensitivity such that a change in blackbody temperature of 6.5 millikelvin amounts to a change in pixel intensity of 1 digital number. So in that scale, this entire video has a median temporal deviation of just 187 millikelvin. So what is there in this temporal deviation? An easy option is to look at the temporal derivative. And we apply a Gaussian pre-filter. We smooth it before computing the derivative. But still, it just looks like noise. And what I'm going to show you is that there was actually a gentle breeze flowing past this plastic bunny that we are able to reveal with our temporal spatial video processing. All this detail was present in the raw measurements, and all we had to do was properly model the physics of microbolometers. There's no learning here. There's no diffusion models here. The key to this all is to analyze the temporal dynamics of the data. So let's take an even simpler scene. Say this plain wall that's at thermal equilibrium. And we point, there are 2 identical thermal cameras here. The second one is having the internal filters of the manufacturers turned on, and it's used just for reference. The raw video from the scene looks like this. You have some optical vignetting, but other than that, the scene is relatively at uniform temperature. If you inspect a particular pixel intensity across time, it's relatively horizontal. And of course, it jitters. There is noise, right? You can use the autocorrelation function to analyze its temporal dynamics. If it was purely Gaussian noise, it would fall completely within this yellow band. But for this function, you see that the autocorrelation function stays above it. Maybe the wall is not at perfect equilibrium. Maybe there are small variations. So we looked at its neighboring pixel. The neighboring pixel is also having low frequency trends. And most importantly, you see that there is this constant offset between them, or some kind of baseline difference between these 2 pixels. This cannot be in the scene. Heat conduction would prevent it. This has to be in the sensor. So what is this sensor? What is happening? It's not a CMOS sensor. Image formation in these microbolometers, and we're not talking about this $100,000 cool thermal cameras. We're talking about cheap microbolometers that you can get even for a few $100s. They are fundamentally different from CMOS sensors. Each of their pixel is a temperature-dependent resistor whose temperature cannot be reset instantaneously, unlike CMOS, where you can just reset the charge. And just the mere act of measuring the pixel resistance alters its temperature. To further add to this, the components of the camera are itself at room temperature. They're itself emitting radiation. The manufacturers have a suite of signal processing algorithms baked into these cameras. This is the video that looks after you have the internal filters on. It looks significantly more stable compared to the raw video. There's less stripe noise. You don't see the striping as much. But if you inspect the pixel plots, the Gaussian noise has been removed, but the neighboring pixels are still not on top of each other. And if we zoom into any particular region, say this region, you see this grainy artifact. You see some grainy motion. And if you keep your attention on any one of these regions, you will notice that some of the pixels are constantly colder or warmer than its surroundings. It is this pixel-to-pixel differences in the temporal drift of these pixels that appears as spatial noise. These are constantly changing due to operating conditions, and this is not something you can calibrate out in factory. It changes based on the history of the sensor, what's seen is seen, and so on. And our basic claim is that we shouldn't be using spatial processing to address this temporal effect. Now that we understand the source of the problem, let's go back to our bunny scene. In this, and let's look at its pixel plots. The first thing we need to do is to apply a temporal 1D wavelet transform at each pixel independently. What that gives you is that it converts these n pixel intensities into a series of coefficients at multiple temporal scales. And the beauty is that the approximation coefficients that is in the first level, that contains all the slow frequency trends. So each pixel's drift gets accumulated into the approximation coefficients, and the detail coefficients are devoid of this drift. So let's look at what happens spatially. Spatially, one of the approximation coefficient maps looks like this. It looks similar to one of the input frames. It contains both signal and the drift. When you come to the detail coefficients, notice that they are on the order of 25 when the pixel intensities are on the order of 20,000. So it's like a tiny, subtle, tiny variations that are happening. But spatially, you can clearly see some spatial structures that were not there in any of the input frames. So these spatial structures, it's noisy, but there's clearly spatial structures. And these come up only after you apply the temporal wavelet transform. And now that we have this, we know how to do image denoising. We have tons of methods. In our case, because of this low SNR, we had to do a custom destriping algorithm, and then we have a off-the-shelf denoising algorithm like BM3D. But you can use whatever spatial denoising you want or build your own. The approximation coefficients require a little extra step because, remember, we have this drift component, so you have these offsets. So when you think about approximation coefficient, you have to remove the fixed pattern noise. So what we do is we apply the same spatial smoothing and look at a mean residual. This becomes a good estimate of the fixed pattern noise. And you can subtract this from the initial coefficient maps. This gives you an initial estimate. And then you can do a second pass, and you get smooth approximation coefficient maps. And that's it. You now know how to denoise every coefficient map, both approximation and detail coefficients. And if you go back and look at the pixel plots, notice that the blue pixel is always on top of the orange pixel. But after spatial processing, the blue comes below the orange. It turns out this particular pixel has actually drifted further apart from its neighbors. And the spatial processing was able to recover it back to its correct position. And you can see the huge level of difference in terms of the input noisy measurements and the denoised measurements. Overall, we call our framework as temporal spatial video processing. So given a noisy input thermal video, you have to apply a per pixel temporal transform, get coefficient maps. You can spatially denoise these coefficient maps however you want, and then reconstruct the video. The focus is to do first temporal and then spatial. And the essence of this is that through this method, we can get videos that are spatially coherent at all times and all temporal scales. The temporal transform itself doesn't distinguish between noise and signal. It just decomposes that intensities into different frequency bins. It is only the spatial processing. So whatever is spatially coherent is signal, and what's not is noise. Now that we have smoothed videos, we can use simple pixel operations to visualize different types of heat transport. We already saw how temporal derivatives are very good at allowing us to see convection phenomena like wind movement. But you can do another option, which is called cumulative change. What we just do is we subtract one of the reference frames from the entire video. What this allows you to do is to see the cumulative effect of some phenomena, like the total amount of cooling that has happened due to a wind passing by. So the scene gets cooler overall. Another option is what we call isocontour maps. And this simply means instead of we apply a color map to the modulus operator. So you take the image, you apply a modulus operator, and then you apply color map. What this introduces is these high contrast edges, which is easier for our perception to actually track. So coming back to our study room, we saw about the bunny. But there are many more types of heat that flows around us, from absorption to conduction, convection, and the different dimensions of radiation. Let's walk through some of these examples. In this case, we have a book and a keyboard that is front illuminated by a white LED panel. The surfaces absorb light based on its color and geometry and warm up. But the noisy temporal derivative is simply incorrigible. But after applying temporal spatial video processing, we get spatially coherent, consistent derivatives, so much so that you can actually read on the book. And the derivatives actually get smoother over time. That's because once conduction kicks in, the contrast will reduce. And if you look at any particular pixel plot, you can see that the noisy measurements are consistently apart, and the denoising makes it smooth, aligns them together, and the profile is also smooth. Next, let's see an example of heat conduction. In this case, we have a black bunny that is illuminated by a incandescent bulb from the right. The input video itself, it looks like almost noiseless. That's because of the high dynamic range of the scene. But once you apply like temporal derivative, you start to see all the amounts of noise in it. And if you perform denoising with our method, then you see consistent effects, and you even see these low, smooth shading effects that is due to the geometry of the object. And the contrast reduces again with time because heat conduction kicks in. For heat conduction specifically, isocontours are a better job. So in this case, if you see in the noisy videos, OK, it takes a bit of time. So you can actually see the noisy isocontours and is completely incorrigible, but here you see these smooth textures. Let's actually zoom into one of these regions, the face of the bunny. The video is playing slow for some reason. Let me redo it. Yeah. So actually, when the video is playing in the noisy video, you feel like you can see the movement, but in any one of those frames, it looks as noisy. It was just our perception being able to track these movements. So if I go to any one of these frames, it's harder to see the motion. But in with our denoising, in every frame, you can see the fine structures of how heat is moving. Our next example is on heat convection. Let's look at this outdoor scene. And let's say we focus on the two cars that are stationary. You can see some motion in the background, and the edges you can see. So you can see the motion-induced edges in the temporal derivative, but nothing else. But once you apply our method, you can actually see that there is vehicle exhaust plumes or the airflow passing through the radiator that becomes visible. That's because these are very slow frequency trends that only the smoothing is able to provide you. Taking further, you can look at thermal polarization, for example. In this case, you only have a bowl of lukewarm water, and you have some reflective surfaces around it. And if you look at the thermal, and we have a wire grid polarizer in front of it that we are rotating manually. And this is how the raw thermal video looks with contrast enhancement. And the temporal derivative, equally noisy. Some of the high contrast edges in the main object is probably seen, but none in the reflections. But once you do our processing, you can clearly see rich amounts of detail, not just on the main object, but also on multiple reflections of it. And if we again zoom into one of these reflective objects, you see rich structures in every point. And if you look at any particular pixel plot, you notice the familiar variation of intensity with angle. In the interest of time, I'll skip this next example. I've showed you like 5 examples in just this scene, but I haven't spoken about photochemistry or tire friction or Joule's heating, for example. And there are many more such things that are happening all around us that I hope this fundamental shift in processing perspective can reveal. And with that, I'm happy to take questions.
We'll have time for at most one question. And if the next speaker could already set up, that would be great.
Yeah, actually quite curious, like what if we just like have like sudden change of like environment temperature when you like open the refrigerator, something like that?
Yeah, so that'll actually be a sharp change in temperature, right? And you'll see all of that. It's just that spatially it'll be incoherent. And with this processing, you can see the rich spatial detail out of that. So any changes.
Yeah, very interesting. Okay, let's thank the speaker again. Thank you so much. Okay, so we have finally the last talk of today. Hold on, before the reception. So this is about ultrasound tomography of the musculoskeletal tissues with generative neurophysics. The presenter is Zhijun Zeng from Tsinghua University. Let's welcome.
Hello everyone, I'm Zhijun Zeng from Tsinghua University. So today I'm very happy to and be so lucky to be the last one to share with you our recent works on how to leveraging the networks and the generative model to achieve quasi real-time ultrasound tomographies of musculoskeletal tissues. So ultrasound tomography is an innovative ultrasound imaging modality which combines the safety of ultrasound and tomography principle of X-ray CTs. So just in last month, the MyJourney, major players in diffusion-based image generations, just announced their entries into the ultrasound tomography, and with their plan to deploy more than 50,000 3D scanners for medical diagnosis by 2031. And I think luckily we may be among the first group to connect the generative model with ultrasound tomography and demonstrate their benefits. Here is the basic principles of ultrasound tomography. We place an annual array around our target, and each transducers will activate as a source, and the remaining transducer will record the transmitted and the scatter signals, and which forms a source to receiver matrix in the frequency domain. So our task is to map these sinogram-like data back to the anatomical structures of the median. So the quality of the imaging depends entirely on what physics we used. In traditional handheld ultrasounds, we use beamforming. So this is sound that wave scatter in waves travels in straight lines. So we detect echo to 5 boundaries, and this gives us only qualitative details, but lacks quantitative detail. So even if we account for band array, we still miss the detail inside the tissues. So to see these deep tissues, we must account for multiple scattering, which requires to solve a nonlinear inverse problem. So mathematically, this is a partial differential equation constrained inverse problem governed by the Helmholtz equations. So we are trying to solve for a inhomogeneous cell speed distribution given the boundary measurement of the wave fields. So this is the joint inverse problem. We have to inverse both the Southby and the full wave field. In fact, in geophysics, the FWI is the gold standard. They use strips to carry air guns to emit wave, and they detect the reflective signals. And FWI allow them to map the deep structures like the gas pocket, the rivers, which is totally invisible if you use ray-based assumptions. So, however, there is a catch. So, solving the partial differential equation is extremely time-consuming, and in clinical diagnosis, we cannot afford to wait days. So, this brings to our solutions. So recently, we are trying to exploring some technique about how we can combine the advantage of neural networks efficiencies and PDE modeling's accuracies together to achieve a quasi real-time FWI in biomedical applications. So this technology is called the neural operator, which just trying to learn a surrogate model from the parameters to the solutions. And it is already shown promised in fluid mechanics, and compared to traditional numerical solvers, it is generalizable, it is fast, it is stable. So, but however, if you want to apply this technique to our high frequencies, strong scattering wave equations, it is still have two unsolved challenges. The first challenge is the simulation to real gap. So in biomedical imaging, we do not have pure data. We do not have enough pure data. So existing researches they use is simple medians like the Gaussian random fields, the simple shape data, which looks nothing like the complex real data in clinical diagnosis. So if we train on this data, the network will completely fail when we apply this to clinical diagnosis. However, to make deep learning work for medical imaging, we need massive realistic training data. But this is a chicken and egg problem, because if you need data, you need efficient solvers. If you want to build efficient solvers, you need enough data. So to break this loop, we built a pipeline called cross-modality digital phantom generations. So since we don't have enough high-resolution ultrasound data, instead we start with the existing X-ray CT or NRI data, because this gives us real anatomies. And we use a vision foundation model called the segment anything model to perform the segmentations. And then we, based on the serological research, we assign the accurate Southby properties to the bones, the fat, and the muscles. And then we gives us hundreds of digital organs, but it's not enough for modeling wave equations. So here's the trick. We do not use this data directly to train our network. Instead, we fine-tune stable diffusions using our segments map. And by carefully design the prompts, we can generate infinite variety of phantoms with real anatomies. And we ask our doctor collaborator to check some of these images. They cannot barely distinguish between the real CT scans and generated phantoms. And this results into more than 30,000 realistic digital organs, including breasts, legs, and arms. And this foundation lays allows us to train our neural networks to learn real biomedical physics. And we then place these phantoms into a digital instrument, a simulator that perfectly matches our physics hardware. So we ran massive wave simulations to create more than 50 million training pairs. And this is largest data set of this kind. And however, even with this data, there's another challenge is that the standard neural networks, they tend to learn low frequency features. But in our cases, the high frequency oscillations are critical. So we need our neural networks to capture the detailed oscillations. So to accomplish this, we propose a the strong scattering neural operator. It is an innovative architecture that incorporates the learnable scattering mechanisms. So just like the physics principle that photon scatter sequentially, we design our networks to model the iterative scattering process. So let's look at the math part. If we want to solve the Helmholtz equations, we start with the inhomogeneous Helmholtz equations. We do an operator splitting, and then we apply the green operators on both sides. This gives us the traditional Born series. It is an iterative scattering mechanism. However, this mechanism diverges for strong scattering media like Borns. So instead, you can start with a damped Helmholtz equations, and then you still do the operator splitting, and then you apply a left preconditioners on both sides. You will obtain the convergent Born series, which is converge for arbitrary large domain or strong scattering. So our network is just mimic this convergent Born series on the latent space. And as a result, the strong scattering neural operators reconstruct the high frequencies interference pattern almost perfectly, and far better than existing deep learning solvers. And more importantly, look at the speed. So for a FWI, our S2NO based inversion is roughly 15 times faster than the traditional numerical solvers. And with parallel computing, this makes the quasi real-time FWI imaging in biomedical possible. So it means that half an hour waiting, you can obtain your radiological results. So let's look at the in vivo breast UT results and take a look at the diagnosis value. Here we compare our S2NO with the traditional delay and sound and time-of-flight tomography methods for the breast UT. So our method clearly resolved the malignant tumors. Here it has the higher speed of sound and anomalistic boundaries, which is a clear sign of tissue invasions. And look at the benign cysts in contrast, they have low sound speed and normally boundary. And this differentiation is critical for early cancer scanning. So next we compare our UT method directly with 3 Tesla NRI, and we found that the agreement is striking. So we achieve a resolution about 1.2 millimeter, and which is comparable to NRI but error fraction of const. Next, we are not limited to 2D, so we build a system where the ring array moves vertically at the water tank to obtain a 3D scan of a human's lip. So you can clearly see the muscle groups, the firmer bones, and even the vessels. So yeah, at this time, our annual ring moves vertically. Yes. And this is the 3D orthogonal views of another volunteer's legs and arms. And finally, we're also scaling our system down to a small animals imaging that supports biological research. So here is our setup scanning a mouth. Yeah, and these are results of a whole body mouse imaging where we resolve internal organ structure with high contrast and opening doors for preclinical drug abuse. Thanks for our collaborators, Professor Hersun from Peking University and Yubin Li from CAS, and my mentor, Professor Zhou Yangshi and Dr. Ho. Yeah, thank you for your listening.
Thank you. So we have time for one question. Do you have any question from the audience? Actually, I'm quite interested because does this kind of like image information model and also neural operator could be migrated to like optical wave imaging, because optics is also kind of waves, right, rather than acoustics.
Oh yeah, we already built a kind of system on optical diffractional tomography, so it works for like all of the wave imaging phenomenon. So the paper will be released soon. Yeah.
Great. Thank you. Okay, let's send the speaker again. Thank you so much. That basically concludes the end of the session.